Imagine intentionally turning off your immune system right in the middle of, like, a deadly viral outbreak.
Right, which sounds absolutely insane.
Yeah, I mean, it sounds like biological suicide. You're essentially just rolling out the red carpet for an invader to hijack your cells, replicate, and, tear you apart from the inside.
Oh, totally. It's the worst possible strategy you could think of.
Exactly. But if you look at one of the most notorious superbugs on the planet, Staphylococcus aureus, you find out that this apparent death wish is actually, well, it's a brilliant, deeply hidden evolutionary strategy.
It really is.
So, welcome to our deep dive into the microscopic battleground of antibiotic resistance. Today we're unpacking a fascinating paper from Nature Communications that completely rewrites how we understand bacterial warfare.
Yeah, and the mission today is to completely unpack how bacteria share their weapons.
Right, like antibiotic resistance. We know horizontal gene transfer drives the spread of resistance, but this research reveals something wild. The bacteria doing the heavy lifting in this arms race are actually the ones with broken immune systems.
Yeah, the underlying paradox here, it changes the entire paradigm of microbial survival. We're looking at a system where a bacterium's supreme vulnerability to a virus is the exact mechanistic trade-off required to survive a much larger threat.
A population-ending threat, right, like an antibiotic strike.
Exactly. Sometimes being weak to a virus is the only way to survive the antibiotic.
That is just so counterintuitive. So we need to pull apart the mechanics of this. Because if you're a bacterium staring down a lethal dose of, say, erythromycin, your traditional vertical evolution, waiting for a random genetic mutation to save you is way too slow.
Oh, way too slow. You'd be dead.
Right. You need a new weapon immediately, which means you have to steal it through horizontal gene transfer, or HGT. But S. aureus operates under a massive biological constraint compared to other bacteria.
It really does. So many bacteria rely on a mechanism called transformation.
Which is basically environmental scavenging, right?
Yeah, exactly. When a neighboring cell dies and bursts open, its DNA just sort of floats freely in the surrounding environment. Competent bacteria can simply absorb this naked DNA right through their cell membranes and integrate it.
Just vacuuming up dead neighbor parts?
Pretty much. But S. aureus, however, is not naturally competent. It completely lacks the biological machinery to just pull free-floating DNA from the wreckage of its neighbors.
So scavenging is completely off the table for them. That forces S. aureus to rely on active directed transfer. And the paper outlines two main physical routes for this. The first is conjugation.
Right, which requires physical contact.
Yeah, I picture this as like a microscopic handshake. A donor bacterium extends this specialized appendage called a pilus, physically docks with a recipient cell, and transfers a conjugative plasmid.
And the researchers specifically tracked a plasmid called PG01 in this study.
And plasmids are incredibly efficient, right? They're these tight circular loops of DNA completely separate from the main chromosome. They act almost like biological USB sticks packed with extra survival traits.
That's a perfect analogy. Conjugation is highly effective, but because it relies on direct cell-to-cell contact, its range is super limited.
Right, you have to be right next to the guy.
Exactly. So if a bacterium needs to acquire a trait from a distance or across a hostile environment, it relies on the third route, which is transduction.
Ah, the viral hijack.
Yes, bacteria are locked in this internal war with bacteriophages, which are just viruses that specifically target and infect bacterial cells.
Right, and these phages, they operate like tiny lunar landers. They attach to the bacterial surface, punch a hole in the membrane, and literally inject their viral genome.
Right, and normally this triggers the opolitic cycle,
where the virus hijacks the bacterium's internal factory, forces it to manufacture like hundreds of new viral copies, and then ruptures the cell to release the swarm.
Exactly. But the assembly process inside that factory is chaotic. Sometimes, as the new viral capsids are being loaded with DNA, the machinery makes an error.
A very happy error for the bacteria.
Yeah. Instead of packaging the viral genome, the phage accidentally packages a fragment of the host bacterium's chromosomal DNA.
So when that misloaded, defective phage bursts out and infects the next bacterium, it isn't actually injecting a lethal self-replicating virus.
Right. It's injecting a package of bacterial genes stolen from the previous host.
And what blew my mind in the sources is that some genetic elements have evolved to do this intentionally, not just by accident.
Oh, absolutely. The paper goes really deep into mobile genetic elements, specifically PICIs.
Phage-inducible chromosomal islands.
Yeah, and they highlighted one called SAPABOV1. If the normal bacteriophage is the hijacker, This PICI sounds like a genomic parasite feeding off the original parasite.
Wow, a parasite of a parasite.
Yeah. It just sits quietly embedded in the bacterial chromosome doing absolutely nothing until a specific helper phage, like the 80 alpha virus, attacks the cell.
And the molecular choreography there is just stunning. When 80 alpha infects the cell and starts building its own viral structural proteins, the sep above 1 sequence wakes up,
It actively steals those newly synthesized capsid proteins.
effectively hijacking the 80 alpha assembly line.
Exactly. It forces the machinery to package the PICI DNA instead of the viral DNA. The resulting burst of viruses is just overwhelmingly filled with these mobile genetic elements.
Essentially weaponizing the virus to spread the bacterium's own specialized traits to neighboring cells. But that brings up a really critical mechanistic question.
Which is?
Well, if these viral delivery systems, whether they're accidental packages or intentional PICIs, involve a virus punching into a new cell, the recipient has to actually survive the encounter to use the new DNA. I mean, a dead bacterium can't be antibiotic resistant.
That is the key point. That survival hinges on the nature of the phage itself. Not all phages immediately destroy their hosts. Some are temperate phages, which utilize the lysogenic cycle.
Okay, break that down for us.
So when a temperate phage injects its DNA, it produces an integrase enzyme. This enzyme finds a specific attachment site on the bacterial chromosome, cuts the DNA, and seamlessly splices the viral genome right into the host's DNA.
So the virus essentially goes dormant.
Right. It becomes a prophage.
And the bacterium just continues to live and replicate, copying the dormant viral DNA right along with its own.
Exactly. This is lysogenic conversion. The temperate phage doesn't just sit there as dead weight, though. It often brings new traits, like accessory genes.
Right. In the study, they note phages bringing along the gene for erythromycin resistance.
Yes, so the host bacterium survives the infection, integrates the dormant virus, and wakes up with a brand new, highly effective resistance profile.
Okay, so let's do a quick 5-minute recap for you listening at home because we just covered a ton of ground.
Sounds good.
So HGT is how bacteria share traits. The 3 main methods are transformation, which is scavenging, conjugation, the physical handshake, and transduction, the viral hijack. And mobile genetic elements are the vehicles making it happen.
Exactly.
So here's a question for you to think about. If you were a bacterium under attack, which of these 3 methods would you rely on to quickly steal a defense mechanism from your neighbor?
I'd probably go with the viral hijack.
Yeah, seems the fastest. Okay, so mapping this out, we have this microscopic arms trade happening through physical Paley bridges and hijacked viral landers. Between conjugation plasmids and temperate phages, bacteria have an incredibly robust system for rapid-fire gene sharing.
They do.
But if this system is so effective, why isn't every single S. aureus cell on Earth totally resistant to every drug we have? Like, why aren't they just one monolithic superorganism freely passing immunity back and forth?
Well, because maintaining an open-door policy for foreign DNA is a massive evolutionary gamble.
Right. You can't just let anyone in.
Exactly. If a bacterium accepts every incoming plasmid or phage, sure, it will occasionally gain a useful resistance gene, but it is statistically far more likely to absorb a lethal Paleolithic bacteriophage that will just obliterate it.
So to manage this existential risk, S. aureus populations are strictly compartmentalized into distinct lineages, right?
Yes, called clonal complexes or CCs.
They operate like microscopic tribes. Transferring genetic material within a single clonal complex is generally accepted, but crossing the border into a different clonal complex is almost entirely shut down by the bacterial immune system.
And the researchers focused heavily on the type I restriction-modification system, the RM system.
Which acts as the ultimate border patrol.
Yeah, the mechanism of the type I RM system is ruthlessly efficient. It relies on 2 opposing enzymatic actions modification to protect the self and restriction to destroy the non-self.
Okay, so I picture this as the security team at an incredibly exclusive, highly paranoid nightclub.
I like that.
The bacterium itself is the nightclub. The VIP guests are the bacterium's own local DNA, and the unwanted crashers are foreign viral genomes.
Right.
Inside this club, you have a 2-part security apparatus. First, you have the stamper. These are the hsDM and hsDS genes.
The modification system.
Right. The hsDS gene acts as the targeting system, recognizing very specific DNA sequences, while the hsDM gene provides the physical modification. It basically walks up and down the genome, attaching a methyl group to specific adenine or cytosine bases.
It's essentially slapping an invisible ink VIP wristband on the bacterium's native DNA, proving it belongs to that specific clonal complex.
And critically, every clonal complex has evolved a completely unique methylation pattern. So the molecular shape of the wristband in, say, clonal complex 1 is fundamentally different from the wristband in clonal complex 8.
Which is exactly where the second half of the security team comes in the bouncer.
The hsdR gene.
Yes, the hsdR gene. It's a highly aggressive restriction endonuclease. It acts as a pair of molecular scissors, constantly patrolling the intracellular space.
Looking for intruders.
Right. Whenever a piece of DNA enters the cell, the hsdR bouncer scans it. If that DNA lacks the exact mathematically perfect methylation wristband for that specific clonal complex,
the hsdR enzyme binds to it and systematically chops the DNA backbone to pieces. Just shreds it.
the empirical data from the study really proves how strictly these borders are enforced. The researchers tested 27 different S. aureus strains across 8 different clonal complexes.
And they attempted to force the transfer of classic phages, right? Like 80-alpha, Phi11, and Phys-LT, along with various plasmids between entirely different CCs.
And the bouncers did their job. Every time.
Yeah, when a phage generated in a CC8 strain injected its DNA into a CC1 strain, the CC1's hsdR restriction enzyme instantly recognized the missing methylation pattern.
The rate of successful horizontal gene transfer dropped by more than 100-fold compared to transfers within the exact same clonal complex.
100-fold. The foreign DNA was just shredded on entry.
The defensive network was functioning perfectly. Plasmids and temper phages were overwhelmingly localized to their specific lineages.
Okay, time for another quick 5-minute recap for you. So, bacteria live in exclusive clonal complexes. Type IRM systems use hsdR scissors to cut foreign DNA, and hsDM and S tags to protect self-DNA, so inter-CC genetic transfer is highly restricted. So, a question for you listening. If these borders are so strictly closed by the bouncers, how does antibiotic resistance manage to spread globally across entirely different lineages of S. aureus?
That is the million-dollar question. The resistance is moving across the clonal complex borders in the real world, so the bacteria clearly have a way to bypass this incredibly tight restriction system.
And the researchers isolated the exact loophole. While classical small-scale genetic elements were blocked, 2 specific transfer mechanisms bypassed the security net almost completely.
Generalized transduction and lateral transduction.
So lateral transduction, or LT, is a massive deviation from the normal viral hijack. Instead of a phage accidentally packaging a tiny fragment of host DNA after it finishes replicating,
Lateral transduction triggers packaging while the bacterial chromosome is still undergoing active replication.
right, the phage starts spooling massive, continuous chunks of the bacterial chromosome. We're talking hundreds of thousands of base pairs straight into its capsids.
The sheer volume of genetic material transferred is unprecedented. But the true brilliance lies in how that material behaves once it enters the new, heavily guarded recipient cell.
But wait, if a massive chunk of CC8 chromosomal DNA gets injected into a CC1 cell, it still doesn't have the CC1 methylation wristband. The hsdR bouncer should immediately attack it.
It absolutely does attack it. The restriction endonuclease binds to the incoming chromosomal DNA and begins severing the strands. The incoming DNA is physically chopped into linear fragments.
Okay, so it gets shredded.
It does.
Yeah.
But the mechanism of integration for chromosomal DNA is fundamentally different from plasmids and phages.
Because plasmids and phages rely on site-specific integrases, and those integrases demand structural perfection.
Right. The incoming plasmid must exist as a pristine, unbroken circular loop. If the hsdR bouncer makes even a single cut anywhere on that loop, the geometry fails.
The integrase enzyme cannot function, and the plasmid is permanently degraded. The defense system wins.
Exactly. But chromosomal DNA transferred by lateral transduction doesn't use integrases. It utilizes RECA-mediated homologous recombination.
Oh, wow. Okay, what is that?
So this molecular machinery doesn't care about pristine circular geometry at all. RICA is a protein that scans broken linear DNA fragments for sequence homology, basically areas where the genetic code matches the host's existing chromosome.
So when it finds a matching sequence, it physically invades the host DNA double helix and literally stitches the broken incoming fragment directly into the host genome.
It's bypassing the shredder by literally patching its own severed pieces right into the host's blueprint. The bouncer cuts the DNA, but because it's massive linear chromosomal material, those fragments just reassemble themselves through sequence matching.
So the restriction system's cuts become completely irrelevant.
Entirely irrelevant. Lateral transduction allows for incredibly high-frequency transfer of chromosomal traits, like the SCC-MET cassette that confers methicillin resistance, straight across the strictest clonal complex borders.
That is wild. But lateral transduction wasn't the only exception the researchers found, was it?
No, it wasn't.
No.
When they looked at the raw transfer data, there was a glaring anomaly involving two very specific strains of clonal complex 1.
Strains JP9179 and JP9181.
Right. While every other CC1 strain in the study was successfully deploying its bouncers to shred foreign plasmids and normal phages, these two specific strains were completely promiscuous.
Meaning they were readily accepting intact plasmids and classical phages from entirely different lineages.
Yes, they were absorbing everything without putting up a fight.
Okay, why?
Well, when the researchers sequenced the genomes of JP9179 and JP9181 to understand this structural failure, they found that these naturally occurring strains possessed a critical mutation. They had suffered a deletion in the genomic region encoding the hsdR gene.
They lost the bouncer.
They completely lost the bouncer. The restriction endonuclease was gone, meaning there were no molecular scissors to cut the foreign DNA.
But the crucial detail here is that the hsDM and hsDS genes, the stampers, were completely intact and fully functional.
And the implications of that specific genetic state are just profound. Because the hsdR endonuclease is missing, a foreign phage from a different clonal complex can inject its DNA, and the cell mounts zero restriction response.
The DNA just hangs out in the cytoplasm unharmed.
Right. But because the hsDM and hsDS modification enzymes are still actively patrolling the cell, they encounter this naked foreign DNA and execute their default programming.
They put the local VIP wristband on the invader.
Exactly. The stamper enzymes physically methylate the incoming foreign phage or plasmid, applying the exact CC1 modification pattern.
So the foreign DNA is perfectly disguised as native CC1 material.
Yep.
Okay, let's do our third recap. So lateral transduction bypasses immune cuts by stitching broken DNA pieces together using regay. And specific mutants lack the hsdR scissors but keep the methylation tags, making them immune-deficient but highly receptive to foreign DNA.
That sums it up perfectly.
So listener, think about this. If a bacterium is missing its primary immune system and is completely vulnerable to viruses, shouldn't it just die out? Why would nature keep these weak mutants around?
It's such a great question. Because tracing this logic, we have a natural mutant entirely missing its immune restriction scissors, leaving it totally vulnerable to any incoming virus, but it retains the ability to perfectly tag foreign DNA as its own.
This sets up the ultimate gateway mechanism.
Yes. To prove this gateway effect, the researchers executed an elegant multi-stage experiment. They isolated a phage generated in a CC8 strain.
Okay.
They introduced that foreign phage to the mutant CC1 strain, JP9179. Just as the genetics suggested, the mutant offered no resistance, absorbed the phage, and its still-functioning modification enzyme tagged the phage with the CC1 methylation pattern.
So they took a weapon forged in CC8 and the mutant polished it up and stamped it with the CC1 seal of approval.
Exactly. Then they took that newly tagged phage and introduced it to a completely normal, heavily defended CC1 strain, JP9197.
And this normal strain possessed a fully operational hsdR restriction system, right?
Yes, fully operational. But when the phage injected its DNA, the normal strain accepted it completely.
Because to the normal cell's bouncer, the incoming DNA was perfectly methylated. It had the correct wristband.
Right. The mutant essentially acted as a laundering operation. It imported foreign, restricted DNA, disguised it with the local modification tags, and then successfully transmitted it to the rest of the highly guarded clonal tribe.
It serves as an interclonal evolutionary hub, but is this just like a neat lab trick?
No, the researchers wanted to know the exact same thing, so they analyzed massive global genomic databases of S. aureus sequences. And they discovered that roughly 4% of all sequenced genomes in nature carry this exact hsdR deletion.
4%.
Yeah, these promiscuous immune-deficient mutants are ubiquitous, scattered naturally across every major lineage.
I mean, 4% might sound small, but in a localized population of trillions of cells, that represents a massive, dedicated subpopulation operating purely as genomic gateways. But this brings us back to the ultimate evolutionary paradox we asked about earlier. If 4% of the population has no functional restriction system, they are hopelessly vulnerable to lethal myeloid bacteriophages. In the brutal mathematics of natural selection, those strains should be rapidly wiped off the map. Why does the broader bacterial population maintain this weak flank?
Well, the researchers answered that with a definitive time-kill experiment, setting up a brutal evolutionary crucible. They created a mixed culture containing a 50-50 ratio of the weak mutant strain, JP9179, and the normal, highly defended strain, JP9197.
Okay, 50-50 mix.
Right. They expose this entire mixed population to a temperate phage. But crucially, this invading phage was carrying the resistance gene for the antibiotic erythromycin.
So they introduced the ultimate dual threat, a viral infection carrying a chemical shield.
Yes. And they split this mixed culture into two distinct environments. In the first environment, they added the phage, but no antibiotics. The results followed basic evolutionary theory. The normal strain, utilizing its intact hsdR bouncers, shredded the incoming phages and protected itself. The mutant strain, possessing no restriction system, was systematically infected by the phages.
So the normal strain just outcompeted the mutants. I mean, without the pressure of the antibiotic, being vulnerable to viruses is just a massive biological liability. The mutants took heavy losses.
Oh, massive losses. But in the second environment, they added the phage and simultaneously flooded the culture with lethal doses of erythromycin.
And the dynamic inverted completely.
Completely inverted. The normal, highly defended strain reacted exactly as it was programmed to do, its hsdR. Bouncers immediately recognized the unmethylated phage DNA and chopped it to pieces, neutralizing the viral threat.
And this is the kicker, because it destroyed the phage DNA, it also destroyed the erythromycin resistance gene carried on that DNA.
Exactly.
The normal cell's own immune system prevented it from acquiring the exact tool it needed to survive. So the erythromycin killed the entire normal population.
Right. The mutants, however, had no defense against the phage. They passively absorbed the viral DNA into their chromosomes, but by absorbing the virus, they absorbed the resistance gene.
Wow.
Yeah. Within 8 hours under antibiotic pressure, the normal strain was entirely eradicated and the culture was 100% dominated by the previously weak mutants.
So they sacrificed their viral immunity to guarantee their genetic adaptability. The survival strategy is entirely population-based.
Right, it's a collective effort.
In normal times, the broader aureus population allows the 4% mutant gateways to take a beating from environmental viruses. But when an extinction-level event occurs, like a massive influx of antibiotics, those weak, vulnerable mutants are the only cells capable of easily absorbing the life-saving resistance gene from a neighboring clonal complex.
They survive the chemical strike, and because their methylation stampers are still working, they repackage that resistance gene and distribute it laterally to any surviving normal cells once the crisis passes.
It is a profound division of labor. The normal cells provide stability and viral defense, while the mutant subpopulation acts as the high-risk, high-reward innovators. They ensure the clonal complex never gets boxed into a genetic dead end.
Exactly.
Okay, final recap for you. Mutants act as gateways, converting foreign DNA into local DNA. 4% of natural S. aureus are gateways. And they face a brutal trade-off, highly vulnerable to phages, but uniquely capable of acquiring life-saving resistance during antibiotic pressure.
Spot on.
So, listener, think about how we design treatments. Are we inadvertently selecting for these gateway mutants every time we use an antibiotic?
Because the implications for modern medicine here are just staggering. We are staring down a global crisis of antibiotic resistance in hospital settings, constantly battling superbugs like MRSA.
Yeah.
And this research suggests our fundamental approach might be structurally flawed. When we blanket a patient with a heavy course of antibiotics, we aren't just killing the susceptible bacteria, we're actively selecting for these 4% gateway hubs.
We are pulling the trigger on their survival strategy.
We are, effectively clearing the battlefield of the normal bacteria and giving these hyper-acquirers unrestricted access to absorb and share new resistance mechanisms.
It fundamentally alters the landscape of infection control. We are inadvertently fueling the very evolutionary mechanism designed to defeat our drugs.
We really are. But, by exposing this specific vulnerability, the fact that 4% of the population will accept anything without restriction, we might actually be able to exploit the bacterial network.
Like a Trojan horse strategy.
Precisely. Since we now know that bacteria rely on these 4% gateway mutants to import new traits, we could artificially engineer our own temperate phages. But instead of carrying resistance genes, these therapeutic phages could carry targeted CRISPR arrays or self-destruct sequences.
Oh, wow. So you deploy the engineered phage into the infection. The gateway mutants absorb it immediately because they have no bouncer. Their stampers perfectly methylate the lethal sequence, disguising it as native local DNA.
Wow.
And then the mutant acts exactly as it evolved to act. It acts as the interclonal bridge, highly efficiently distributing our self-destruct codes past the bouncers of the normal, heavily defended bacterial population.
Exactly. We use the bacterium's ultimate survival loophole to bypass its own immune system, forcing them to spread their own destruction to the rest of their clonal complex.
That is incredible. We started today looking at a landscape that felt deeply frustrating, where the classic models of bacterial genetics just couldn't explain the rapid cross-lineage spread of superbugs.
Yeah, it didn't make sense before.
But by digging into the mechanics of lateral transduction and these immune-deficient mutants, we've uncovered the hidden architecture of bacterial survival.
We have.
The bacteria aren't just randomly mutating, they're maintaining an incredibly sophisticated, interconnected risk management network. It's a terrifying reality for hospital stewardship, but as you just said, it's also a brilliantly hackable system. Something for you to mull over next time you hear about a superbug outbreak. Thanks for joining us on this deep dive.